Okay, some additional characteristics that don't behave quite as perfectly as Mendel's characteristics in his P's are polygenic traits. And polygenic traits have multiple genes and multiple alleles, so lots of places across your genome that control it. Some examples of polygenic traits are height and skin color. We're finding more and more different types of autism are also polygenic. And you can easily spot a polygenic trait if there's continuous variation. So there's a wide range instead of just individual categories. So we can see these gentlemen are all slightly different heights. Anything, if you take it and graph it out and get a bell curve, and a bell curve looks like a bell. So you have most of them somewhere in an intermediate, and you have some on either end. Bell curve type traits are going to be polygenic, lots of things affecting them, instead of just green or yellow, wrinkled or round. Those are just two discrete categories rather than all sorts of possibilities throughout. Also, there's pleiotropy, and pleiotropy is where one single gene has an effect on multiple traits or characteristics. An example of that is sickle cell anemia. So if you have sickle cell anemia, you get two copies of a defective blood type gene, and your blood cells look sickle-shaped. Well, people with sickle cell anemia have a lot of health problems and historically didn't survive very long because of this blood type. It's caused by one location in the genome. If you do not carry the sickle cell gene, you're completely normal. If you have the sickle cell gene, you don't have the disease because you have normal red blood cells, but some of your blood cells can change to sickle shape. The second characteristic influenced by that sickle cell gene is malaria. So with malaria, if you have the homozygous characteristic, you're susceptible to malaria. However, if you have the heterozygous characteristic, so hiding those sickle cells, you're more immune to malaria. Your body has an easier time fighting off malaria. If you have actual sickle cell disease, you're immune to malaria, but that's probably not much help because you're very sick already with the disease. So hiding the sickle cell gene is actually the most beneficial condition, even though you have a chance to pass it on to your offspring, especially if you live in an area that has a lot of malaria. Another characteristic that we see in a lot of organisms are genes that are carried on chromosomes that determine gender, or their sex-linked traits. And in humans, to be a male, you get one X chromosome and one Y chromosome, and that determines that you're male. To be a female, you need two copies of the X chromosome. So depending on which of these you inherited from your father, it determined if you are a boy or if you're a girl. Now, if there's a characteristic on that X chromosome, it's going to be inherited differently in males than in females, because females are going to have two copies, and males will only have one copy of that characteristic. And this is true with red-green color blindness. So being able to see normally is dominant, so capital R, and having defective light sensors, so you have that red-green color blindness, is recessive. For a male to be colorblind, you only need one little r, because there's nothing on the Y to cover it up or hide it. For a female to be colorblind, you have to get colorblind from both mom and dad. So it's much more common for males to show colorblindness because it's on the X chromosome. To have normal vision, a male just needs one copy, and a female can have one or two copies of the normal gene. Sex-linked characteristics were first discovered by a guy named Thomas Hunt Morgan, and he was looking at fruit flies. And he wasn't looking specifically for sex-linked characteristics. He was just looking for a natural mutant fly. This was back in the day before we knew how to artificially cause mutations. in fruit flies, so he was looking for a natural mutation that he could use to study in his lab. So here's Morgan and a picture of his lab behind you. And so he would go out to the open fruit markets with nets and capture flies and bring them back to his lab, put them under the dissecting scope, and study them to see if there were any different characteristics. And because natural mutations are fairly rare, he wasn't having much luck. He'd been at it for months and hadn't found any mutants, and he was really studying his flies, even going so far as to count the hairs on their back to see if there was any variation in number of hair follicles that he could work with to cross. So he was really studying these flies. And this is what a fruit fly looks like normally. Red-eyed, wild type, or normal type fruit fly. He'd been looking for months at these things, and that's all he had found. One day he went out and collected, came back to his lab, and started dropping flies on the scope, and he saw this. It was a white-eyed fly. Drastic difference from the red-eyed fly, and he was so excited. He jumped up and he ran down the hall to tell all of his colleagues, "Hey, look what I found! It was an awesome mutation! Come see, come see!" And everybody ran back down the hallway to Thomas's little lab here, and they all got in the room and looked at the dissection scope, which is pictured right here, and in his absence, the fly had woken up and flown away. So this group of PhD researchers quickly closed the windows. This was before there were screens in the windows, so that was wide open to the outside, closed all the windows, and were searching around this little room for one single fruit fly. And there were already a bunch of fruit flies flying around. Anytime you work with fruit flies, some get away, and then they'll buzz around the room for a while. So they're hunting for one tiny little fruit fly in this room, and the room was probably quite uncomfortable. You can see bananas in the background. They would smush bananas to give the little fly maggots something to grow on. They used ether to knock out the flies, make them sleep for a while, so it just probably smelled awful. They're hunting around in the middle of summer, a big group of guys in this tiny little room looking for one fly. They eventually did find it up on a window pane, and he recaptured his little male fly that had white eyes, and he used that to set up his crosses. So I'll walk you through the crosses. Again, the white-eyed fly was male, and he just pulled a red-eyed fly out of the things that he had in his lab. And when you're doing sex linkage, you have to include the sex chromosomes. So XX for female, XY for male, and that's separate from the red and the white. But we're going to use them as superscripts. So he discovered that red was dominant and white was recessive. So red will have a capital R and white a lowercase r, because it's complete dominance and complete recessive. And our white-eyed fly over here, being male, it has an X and a Y chromosome. The female has two X chromosomes. He's red-eyed, or excuse me, he's white-eyed, so he has a little r as a superscript on his X. Leave the Y empty, because remember, they don't match, so the Y is not going to carry a matching gene. And the female that he pulled from his stocks happened to be homozygous dominant, two red eyes. So he did his cross, and I'll do a Punnett square here. Shows our XRY from the male, and our X's with capital R's for the female. So pull the chromosomes and the traits that they carry all the way through. And what we end up with are all of the offspring have red eyes, because all of them have at least one capital R. So all red eyes, half of them are male and half of them are female. Now if we cross one of these females with one of these males in a second Punnett square down here, so now I'm pulling out of the middle of that top square, and I'll cross a male from up here with one of these females, we'll get a female, again, that's homozygous dominant for red eyes. We'll get a male with red eyes. We'll get a female that's heterozygous or carrying red eyes and white eyes, and we'll get a white-eyed male. So that's what we would get with this cross. And we'll get some practice with these on some worksheets in lab. Some other things that can have an effect on what your genotypes do and what phenotype you see are environment. So environmental effects, there's a wide variety of them. We'll just look at a couple. Diet. So diet can have an effect. If any of you have phenylketonuria, that means that your body can't break down the amino acid phenylalanine. So you need to watch out for things like on Coke products contain phenylalanine and not consume those products. This is important because individuals born with phenylketonuria, because they can't break down phenylalanine, if they have it in their diet, it will build up to toxic levels and can cause lots of things, so mental retardation, even death. However, if at birth you know that you can't metabolize phenylalanine, then you can change the baby's diet so that they restrict phenylalanine and they develop normally and have no health problems, except that they really have to watch their diet. So diet can have a huge impact on what actually occurs with phenylalanine. Also, temperature can have an effect. So turtles, their gender is determined by temperature. Whether they're hot or cold determines if they are male or female. Lots of reptiles do the same thing. Fur color in many mammals does this as well. So these Himalayan rabbits down here, they have dark patches wherever they're slightly colder and completely white where their body temperature is warmer, keeping the hair warm. Siamese cats do the same thing. So those darker areas of the fur are slightly cooler regions of the body. The lighter areas are warmer areas of the body. And I've seen lots of Siamese cats. In fact, my aunt had Siamese cats when we were little, and Siamese cats are kind of mean to begin with, and I was always afraid of her cat. I hated visiting my aunt because of this awful cat that she had. What was really freaky about her cat is in the winter, she lived in Colorado, so in the winter months this cat would lay on the heat registers and would only lay on one side and never roll over. So half of this cat's face would get to be completely creamy white, and that would blend into black going across. And it was a really odd looking cat over the winter months. And then it would growl and hiss and charge at us kids. And so it was just a scary cat. But that was caused by temperature. The cat had artificially warmed half of its face, so it turned the lighter color. You can also artificially do this with the rabbits if you, not continuously, but if you put an ice pack on their back and leave it for a while each day, gradually that hair as it grows in will be dark in color. So you can manipulate the color of these rabbits and actually give them dark patches on their back. There's lots of other factors that also can interact with the genes. Another example of temperature would be Arctic foxes. They're light white color in the winter when it's cold, and when they shed and grow their summer coats, it's warmer, so they produce pigment and grow in a dark coat. So there's lots of characteristics controlled by the environment. The last kind of concept we'll look at for this chapter are pedigrees. And pedigrees are used very commonly in breeding plants and animals and things like that, but they're also the only way to look at human characteristics, because you can't do with humans what we did with pea plants, pick a male and a female and put them together, make them have a whole bunch of offspring and count the offspring. That just doesn't work for humans. So for humans you need to use pedigrees. And in pedigrees you can determine if a characteristic is dominant or recessive. You can also figure out if it's X-linked or autosomal. But instead of looking for ratios, like we did in our previous crosses, you look for patterns in pedigrees. And we'll get some practice with pedigrees in class, but some examples of patterns and how to construct a pedigree. First, you use the information to map the matings and offspring for your pedigree. So that's what's shown up here. So we have grandmother and grandfather. They're connected by a line which has lines coming down off of it. So it shows that they've had offspring. Some of those offspring have married and produced their own offspring as well. So you first draw the families out, and then you fill in anyone who has the trait. So in our pedigrees back here, whatever your trait of interest is, doesn't matter what it is, you'd color them in completely. So grandfather, this aunt, this first cousin, this father, and this sister, all relating to this individual, have a characteristic that they're interested in. Then you look for patterns, and the patterns, if it skips a generation, try it as recessive because it might be hiding in some of those individuals. So half shade any who have to carry the gene or be hiding it. And then you can add letters to show the genotypes and confirm that you have the correct pattern. If it's in every single generation, then you need to try it as dominant. And again, add letters at the end to show that that actually works out. If you're not sure on any of them, you can't just guess, you can leave them blank. So if you know that an individual has to have a capital letter, but you don't know if it's got a second capital or a little letter for that second one, just leave an underline for blank. And if the letters don't work out on your first guess, you just have to try again. It's kind of a trial and error type thing. Although pedigrees are the only way to study human genes, there's a lot of problems. So oftentimes the history is unknown for a variety of reasons. Maybe in the past some things just weren't talked about. In the past, cancer was an awful word. People just didn't say if they had cancer. So if you're mapping your family to look at a certain type of cancer, you simply may not know that your great-grandma Ethel had breast cancer because nobody talked about it back then. Also, it's very sadly common that the family member who does know the history dies or gets Alzheimer's or something like that before passing on and recording all of that family knowledge. Other things, even if you do record it, maybe the records are lost just through time, they got misplaced, or maybe there was a flood or a fire and you lost all those records. Another issue could be paternity certainty. It's fairly common and well documented that sometimes the father's not always the father. One case that I know of, and it was a friend of mine in college would do pedigrees for families to check to see if they could potentially pass things on to their offspring, and that's what he was studying for his degree. He currently works at a hospital, and he does pedigrees for a living to look at different characteristics. Like, remember when we talked about Tay-Sachs? So he sees families that have Tay-Sachs to see if the parents could possibly carry it, things like that. But he had interviewed this family and done all the paperwork. He had some really cool computer programs that would help him make nice looking graphs. And he sat down with the family, and when he walked into the room, the parents were sitting on one side of the table, and he just sat down right across from them, and he started explaining the pedigree to them. And he was a very professional guy, so I'm not quite sure what he said, but I'm sure he was very professional about it. One of the children in question, when he got to that child, he had to point out that based on the characteristic in the family, that child had to have a different paternity because they did not have the characteristic that they were graphing out. And when the gentleman across the table caught on to the fact that that wasn't his kid after all, instead of turning to his wife and saying, "Honey, can you tell me about this?" he attacked the geneticist who was going over the charts. He jumped across the table and took a swing at the guy that was going over the charts. And my friend said he just turned around and ran out the door behind him, left his charts and everything, never sent them a bill, never contacted the family again. He said it was very sad and kind of scary, but sometimes you just may not know. So along those kind of awful lines, I'll have you guys read a chapter in the Mean Genes book that came with your textbook. And the chapter is called Infidelity or Cheating Hearts. It starts on page 173. The paper must be typed. In fact, you're going to submit it to Blackboard. There's a drop box for it. Write a brief summary of the chapter. So go through the chapter and write a summary of the chapter. Make sure it's in your own words. and full paragraphs, not just bullet points listing what's going on. Also, that's about half the points is the summary. The other half the points is include what you learned. What did you learn? What impact did it have on you? And that's where you're going to get your other half points out of this assignment. Submit that on Blackboard, and that's due on Tuesday.